Dear students, the topic is DNA damage and repair.
Before going to the topic, please subscribe my channel. Press the bell icon for notifications. Like, share among your friends and comment. DNA is a highly stable versatile molecule. Though sometimes the damage is caused to it, it is able to maintain the integrity of information contained in it. The perpetuation of genetic material from generation to generation depends upon keeping the rate of mutation at lower level. DNA has many elaborate mechanisms to repair any damage or distortion. The most frequent source of DNA damage are the inaccuracy of DNA replication and chemical changes in DNA. Malfunction of the process of replication can lead to incorporation of wrong bases which are mismatched with the complementary strand. The damage causing chemicals break the backbone of the strand, chemically alter the bases. Alkylation, oxidation and methylation cause damage to bases. X-rays and gamma radiation cause single or double-stranded breaks in DNA. A change in the sequence of base, if replicated and passed on to the next generation, become permanent and lead to mutation. At the same time, mutations are also necessary which provide raw material for evolution. Without evolution, the new species, even human beings, would not have arisen. Therefore, a balance between mutation and repair is necessary. Next we can see type of DNA damage. Damage to DNA include any deviation from the usual double helix structure. The types of damages are simple mutations, denaturation, missing of bases, chemical modification of bases, then formation of pyrimidine dimers, that is thymine dimers, and strand breaks. The first damage mechanism is simple mutation. You know, simple mutations includes transition and transversion. So what is transition? Transition means the replacement of one purine base with another purine base, or a pyrimidine base with another pyrimidine base. And then transversion means replacement of a purine with a pyrimidine or a pyrimidine with a purine. So this is the simple mutation.
Here shows the simple mutation as transition and transversion.
Next damage is due to deamination. The common alteration of form of damage include deamination of cytosine to form uracil, which base pairs with adenine in next replication instead of with which the original cytosine would have paired. As uracil is not present in DNA, adenine base pairs with the thymine. Therefore, cytosine guanine pair is replaced by thymine adenine in next replication cycle. Similarly, hypoxanthine result from adenine deamination.
This representation shows the deamination of DNA lead to mismatch pair.
Next is missing bases. Cleavage of N-glycosidic bond between purine and sugar causes loss of purine base from DNA. This is called depurination. This apurinic site become non-coding lesion. Next is modification of bases, that is chemical modification of bases. Any of the four bases of DNA lead to modified bases. Methyl groups are added to various bases. Guanine forms 7-methyl guanine, 3-methyl guanine, and adenine form 3-methyl adenine, and cytosine form 3-methyl cytosine. Replacement of amino group by a keto group converts 3-methyl cytosine to thymine.
This table shows the modified base pairs and their functions.
Next is formation of pyrimidine dimers, that is thymine dimers. Formation of thymine dimers is very common, in which a covalent bond, that is cyclobutyl ring, is formed between adjacent thymine bases. This leads loss of base pairing with opposite strand.
Here shows the thymine dimer in one strand.
Bacteria may have thousands of dimers immediately after exposure to UV radiation.
This diagram shows the formation of cyclobutane dimer.
Next is strand breaks. Sometimes phosphodiester bond break in one strand of DNA helix. This is caused by various chemicals like peroxides, radiation, and by enzyme like DNases. This lead to breaks in DNA backbone. Single strand breaks are more common than double strand breaks. Sometimes X-rays, electronic beams and other radiations may cause phosphodiester bond break in both strands, which may not be directly opposite to each other. This lead to double strand breaks. Some sites on DNA are more susceptible to damage. These are called hot stops.
This is a double strand break lead to DNA damage.
Next we can see about repair mechanisms. Most kind of damage create impediments to replication or transcription. Altered bases cause mismatching or mispairing and can cause permanent alteration to DNA sequence after replication. In order to maintain the integrity of information contained in it, the DNA has various repair mechanisms. The various DNA repair mechanisms are direct repair mechanism, excision repair, mismatch base repair, then recombination repair or retrieval system and SOS repair mechanisms. We can see the repair mechanisms one by one in detail. The first one is direct repair. The damage is reversed by a repair enzyme which is called autoreactivation. This mechanism involves a light-dependent enzyme called DNA photolase. The enzyme is present in almost all cells from bacteria to animals. It uses energy from the absorbed light to cleave the carbon-carbon bond of cyclobutyl ring of the thymine dimers. In this way, thymine dimers are monomerized. Next is the excision repair. Excision repair include nucleotide excision repair. Base excision repair system involves an enzyme called N-glycosylase which recognizes the abnormal base and hydrolyzes glycosidic bond between it and sugar. Another enzyme, an endonuclease, cleaves the DNA backbone on the 3' side of the abnormal bases. Then the DNA polymerase by its exonuclease activity removes the abnormal base. DNA polymerase then replaces it with a normal base and DNA ligase seals the region. So nucleotide repair system include 3 steps: incision, excision and synthesis. Incision is done by endonuclease enzyme precisely on either side of the damaged patch of the strand. In this way, damaged portion of the strand is cleaved. Endonuclease enzyme involved are UVRA, UVRB, which recognize the damaged stretch of the strand. UVRC make 2 cuts, that is incisions, on either side. Exonuclease removes the damaged strand. Enzyme involved is UVRD. Later, DNA polymerase synthesizes the new strand by using complementary strand as a template. DNA ligase forms phosphodiester bond which seal the end on newly synthesized strand.
Here shows the direct repair mechanism. One strand has a stretch of damaged DNA. Endonuclease cleaves the damaged portion by incision. Exonuclease degrades the damaged stretch. This leaves a gap in the damaged strand. Then DNA polymerase synthesizes new strand. DNA ligase forms phosphodiester bond which seals the ends.
Next is mismatch base repair. Sometimes wrong bases are incorporated during a replication process. Guanine to thymine or cytosine to adenine pairs are formed. The wrong base is always incorporated in the daughter strand only. Therefore, in order to distinguish the two strands for the purpose of repair, the adenine base of the template strand are labeled or tagged to methyl groups. In this way, the newly replicated DNA helix is hemimethylated. The excision of wrong base occur in the non-methylated or daughter strand. The next is recombination repair or retrieval system. In thymine dimers or other type of damage, DNA replication cannot proceed properly. So a gap opposite to thymine dimer is left in newly synthesized daughter strand. So the gap is repaired by recombination mechanism or retrieval mechanism, also called sister strand exchange.
This diagrammatic representation explains recombination repair or retrieval system. See here, damaged DNA strand with a thymine dimer. Then during replication, gap opposite to the dimer is left in the daughter strand, that is shown here. Then gap is filled by retrieving a stretch from the normal good homologous strand. Thymine dimer is monomerized.
The next is SOS repair mechanism. Sometimes the replication machinery is unable to repair. So the portion and bypasses the damaged side. This is known as translesion synthesis, also called bypass system, and is emergency repair system. This mechanism is catalyzed by a special class of DNA polymerases called Y family of DNA polymerases, which synthesize DNA directly across the damaged portion.
This diagram shows molecular mechanisms for DNA repair.
What if the DNA damage repair mechanism fail? An accumulation of DNA damage is commonly related to cancer or degenerative diseases and usually affects a specific cell type or organ. In contrast, the non-functionality of one or more DNA repair proteins as a result of genetic disorder will impair DNA damage repair in all type of cells simultaneously. As an example, genetic diseases like xeroderma pigmentosum, Cockayne syndrome, trichothiodystrophy exhibit defects in the nucleotide excision pathway, NER. Nucleotide excision pathway generally involved in resolving single strand breaks and pyrimidine dimers, which are primarily inflicted by UVB radiation. Such patients are highly photosensitive and prone to skin cancers. Thank you for watching. See you next class with another topic. Don't forget to leave a like and please subscribe my channel.